what's happening for the rest of the afternoon after the next talk. There will be parallel sessions between 4:30 and 6:30. What these will be convolutional models, which will be in this room. And then there'll be generative models in EBR 30. There will be AI in Kenya in EBR 100. So those rooms we've used already if you've gone to EBR. But now there'll also be data science, which is in EBR 432. Right? So you go into the same building, but you're going to go to the 4th floor for the data science session. Tonight for the evening event, as I said, if you sign up for it, there will be the Indaba Women in Machine Learning, and it's sign up or invitation only. The sign up is at the Microsoft booth. Okay. All right. For this next keynote, we are going to have Prof. Abdoulaye Diallo. He's a professor of computer science and bioinformatics at the University of Quebec in Montreal. He is the director of the Bioinformatics Laboratory at Center of Research Excellence in Orphan Disease at the University of Quebec, and is the member of the Mathematics Research Center of Canada. He's also the co-founder and chief scientist of My Intelligent Machines, a startup integrating genomics, bioinformatics, and artificial intelligence. Yeah, let's give a warm round of applause to Prof. Diallo. Okay, another round as he comes up.
Thank you, everyone, and thank the organizer for inviting me. It's a real pleasure for me to be here. As a Canadian, I just want to advise at the beginning that it will be a bilingual presentation, English and French. The Francophone, let's say, whoa, yeah. it's just a joke, because but don't be disappointed, okay? So I will start my presentation by saying that I'm Abdoulaye Diallo. I grew up in Guinea, West Africa, and I went to Montreal in 2000 to do my bachelor. So I did a bachelor and master at the University of Quebec, then a PhD at McGill, and a postdoc, where actually something happened, because when I was at high school, I hated biology, chemistry, and biochemistry. So that's why when I decided to go to university, I chose math and computer science to be sure to do not have to deal anymore with biology. But now I think I'm stuck with it for the rest of my life. Okay. So why it is important, the biology, and what inspired us in the fields of using computer science, mathematics, artificial intelligence in life science, it's because it's the only way that we can deliver precision in life science. Why? I will explain that to you during the presentations. We know that the population is growing, that's an indicator, but also we need more food, we need better healthcare, better precision, preventive healthcare. All those things will need to have a strong bioinformatics and AI component. So my job is threefold. First, as a genomics specialist, I used to generate a lot of genomics data. I will explain later a bit what genomics data are. And genomics are DNA-based data, or the other forms we will talk about later. They are one of the massive data that can be accessible now throughout the world. And then, having those set of data, the next step will be to mine them and to design sophisticated methods to solve problems that life scientists faces. We call that decoding. We decode by using bioinformatics tools. And the last thing is, you want to deliver that technology to the life scientists, to the doctors, to people who need that kind of tools based on the genomics data. So I think everyone in the room is aware of data science. Data science, yeah, it's the option that is presented here. It's a mixture between domain knowledge, statistics, and computer science. So let's just remove the domain knowledge by a big biology box. Okay? So the domain knowledge will be biology, and it is surrounded by other components of the biology, biochemistry, biology, statistics, and mathematics. It is important to notice that since 2014, it's one of the growing domains which need human resources throughout the world. And there are a lot of papers dealing with it. And in Africa, there are projects to build master's and PhD programs in bioinformatics that are mostly led by H3A Bionet. And in different countries, they are building core facilities in computer science departments or in biology departments in different countries. Knowing that we are at the intersection between biology, computer science, and statistics, actually we are struggled in the middle of two types of individuals. The first one mostly are the ones who generate the data, the biologists. They go to the ground or they are in the lab, they generate samples, and from those samples, they extract what we call cells, DNA, genes that are expressed. They measure different variables within that cells, and they give us those data. Usually, it's terabytes of data depending on the experiment. From this, our job is to build computational model to predict outcomes, gene effects, and the influence of markers for diseases. We deal with the other part where we deliver the services. It is doctors that could decide if we have, let's say, a mutation that has been found in a critical genes, is that genes will conduct us to have an influence for a given disease. I can give you one example that I faced in my career. When I was a postdoc at MIT, I had the chance to have a daughter. She was born in Boston, and she went to intensive care because of health failure. We did genetic tests, and from those genetic tests, we have an answer that she has a predisposition of health failure. But knowing that, we can act in a preventive way during her lifestyle. And we are taking that into account in her growing. So that's the chain where we have the biologist, my job, and people who are informed with the results of the data that we analyzed. Actually, there is a boom in bioinformatics, and there are a lot of companies, startups out there doing a lot of fantastic jobs. I think everyone have heard about 23andMe. No, 23andMe is a genotyping company where they analyze for $50 to $100 your DNA, and they look for thousands of markers for different kind of disease. They look for ancestral relationship, and they even found your cousins that are present in the data bank based on your DNA. Company like DNA Nexus or MIMS, the company that I founded 2 years ago, are specialized on delivering bioinformatics solutions to clinicians and to people who need it. And in the next phase, I will show you why it is complex to deliver such precision using bioinformatics and artificial intelligence. Just to be aware also that even now in clinical facilities, let's say in breast cancer, for instance, you can have a genomic test that can be considered depending on the chemotherapy trial that they want you to get instead of getting another treatment. So you can have better chance to survive depending on the type of genomics data, DNA type that you have, and their impact with the type of breast cancer that you have. So we are entering a new phase, a phase where instead of only using your symptoms that we have found, let's say when the first time I saw my daughter at home, she was just coughing. So the symptoms is coughing, and from those symptoms, the doctors take a cure. We are facing now new kind of treatment where the treatment and the follow-ups are personalized based on the genotype of the individuals. And this leaves us with a tremendous amount of data where we have to combine personal molecular fingerprints, multi-omics data, molecular imaging data, and a lot of big analytics within these steps. The other part of that precision is in drug. I think Aisha mentioned that on her talk this morning, saying that one of the major challenges in drugs treatment is the precision, because most of the drugs have been tested within a small subsample that do not cover all the endotypes that are possible, and maybe they fit only a small group of individuals. Targeting those small group of individuals will help to have a better cure. And in the next generation, we will be more facing to have different type of drugs for different type of individuals based on their different type of DNA. That leads us to notice that we are going from sequencing little data. Actually, when they started sequencing data like the Human Genome Data Project, they spent $3 billion sequencing one genome. But now in my lab, I can sequence one genome for less than $100. So knowing that you can sequence a DNA from an individual for less than $100 open a lot of roads about different type of informations, prevalence of disease, treatments that can be developed and precise for that individuals. In all of the northern country, there are now pilot projects on having DNA sequenced for different individuals in different facilities. For instance, in Quebec, we have more than 200,000 people that are already sequenced, and their genome is accessible there for different treatments. So someone will notice that the amount of genomic data will be higher, and it will face a big data problem. Why it is complex to analyze DNA? Because instead of taking, let's just come back and have, I just want to show you that I have my biology lesson correct, okay? So, at the beginning, when we talk about DNA, we just talk about letter, A, C, G, T, 4 letters. 4 letters that can be derived into words that can be represented maybe as genes, as transcription factor, as regulators. And they are packed within the genomes using nucleosome and so on. I'm not going into the details. The only thing that I want you to remember is for each number that I have in my slide here, there are technologies to access those kind of data and to extract them. And when those data are extracted, they can influence the analysis that people will be doing. So that means we can have in the data, a data from a variation between A, C, G, T in your genome, or maybe I have a gene that is expressed in one condition and someone has a different variant of expression of that gene. There are proteins, there are metabolites, and so on. The role of AI and bioinformatics here is to deal with these heterogeneous type of data, integrate them, find signatures, and associate them to therapeutics. So there are workflows for integrating omics, genomics within pipelines that include a huge step of artificial intelligence in the middle. Because when we extract the technologies, we will have to integrate those data. There, machine learning comes in. Because we want to have that quick, we want the algorithms to be accurate, and we'll be handling massive data. All the other aspects of data science and big data that people are facing in the other areas are present within the fields of bioinformatics. So someone can use AI, different type of representation, to extract, to featurize, and learn from those features how to respond to a treatment or how to classify patient that has good outcome within a drug and bad outcome within a drug. So that will integrate visions because there are a lot of images in the area. We are dealing with electronic health records where people usually take the unformatted health records and transform them into format that can be transposable and shareable within different facilities so that they can be used in clinical facilities. And knowing that we are in a treatment, we will be facing what we call a temporal sequencing events, where we will be looking for prognosis, diagnostic, and prediction in the future. So different layers are out there, genomics, sequencing. So we will need to learn for each different type of nucleotide mutation that are present in an individual sequence. These data are transformed into neural nets, and they are used in different conditions. Okay. So having that step of AI within biotechnologies and omics data will help us to cover the different layers that we have in the fields of precision. Because I think everyone heard about personalized medicine or precision in health. Personalized medicine or precision health will have to face those different layers. The first layers is what we are exposed to, the exposome, so the different metabolites and the different radiation that we have, the reaction of our genomes, the microbiome, the microbes that are related, the metabolites, the proteins, the transcript, the genes that have been transcribed, the modification within the genomes, and layers that are related to the area we are living. So the main challenge is how do we design sophisticated method to integrate several layers within these different layers. For now, some of the data are difficult and really expensive to get, but they will be cheaper soon. However, for layers like transcriptomics, genomics, proteomics, and now epigenomes, we do have several data that are out there. And we will have to interconnect between those different steps. So just remember, we cannot deliver precision without dealing with the molecules. It is important to notice that. We need to deal with the molecules if we want to deliver precision in health or in agriculture. Okay? So that will help us dealing to integrate the different type of data and not to approach the bioinformatics as it used to be a method where we just cluster, classify, and detect new patterns. But we will now need to include them in more sophisticated models where we do have multi-omics data, multiple kernels that can be dealt with different representations of the data so that we can answer questions. Those questions could be like, we do have a molecules, let's say a new drugs, that is designed in a pharma, within a pharma. We can model an AI to test if that drug fits a therapy. This is where we are heading. The other things that we can do, we can take all the multi-omic data and look for the prediction of outcomes regarding disease. And we can use those kind of data also to accelerate the process within the hospitals. Let's say, if we know that someone, the recurrence of her cancer is longer than the other one that could be in a risk, you can plan better the timing, the schedule when he's gonna meet the doctor. Knowing that we are in areas where specialists are rare, having those kind of technology will optimize the interconnections between the doctor and the patient regarding the disease we are facing. However, it's not easy because integrating those data, I think someone told me, and he's one specialist, he told me, life science data is the worst and the most difficult data people will have to deal with. Quote Joshua Bengio. Okay. Because we have to integrate those layers within different hidden layers. We have bottlenecks. We can use the outcomes depending on the segmenting populations, and we can associate that to predictors. But life science is so complex that you cannot only deal with the architecture of your neural net, you need also to capture the expertise of the domain, the biologist, the therapist, and the language of the domains that defines the different outcomes of and the different relationship between the genes. Those need to be captured and integrated within the framework of the artificial intelligence that people are developing. So just a recall, AI can have an impact in different areas in health. I won't go into the details. I think you have another keynote dealing with healthcare, and you will go in details about that. I just want to featurize that the type of data that we are facing are clinical images, EHR, genomics, sensors data, and the type of models that people are building are based on CNN, LSTM, and so on. The opportunities here are really to enrich features that are derived from data, life science data, and also to federate data from different origin. I have a good example in the center that I'm heading the genomic and bioinformatics core. We are focusing on orphan disease within the state of Quebec. Orphan diseases are neglected usually, and what's happened by integrating models to capture the signature of different genes, we can mix therapies from different orphan disease together. So within the same framework, we can have different type of infrastructure to interconnect those kind of data. We are in this center, actually, my lab is generating almost 1 terabyte data every 2 days regarding those projects. Even in cancer, you have AAA negative breast cancer, which is neglected and which is one of the most predominant in West Africa of type of breast cancer, and the therapies are lowered behind. So that gives you the perspective of machine learning, bioinformatics, AI, within the context of multi-omics data. Next, let's just show applications, things that my lab, people in my lab is doing. So first, since it is a complex domain, the first thing that we need to have tools to automate tasks and also to develop a curriculum. Workflows, scientific workflows are built, and from those scientific workflows, what can someone do? We can extract knowledge because we can use scientific literature and to design protocols. And from those protocols, we can design bioinformatics workflows. Those bioinformatics workflows can be directly linked within procedures. Okay. So let's say we have an example of an article we can tackle using the domain expertise. We extract the different labels, and from this we reconstruct the workflows, and from the workflows we can map it into the ontology domains. From that domain, we can run it directly within a pipeline. So this kind of technology is the basis of a startup we found 3 years ago in Montreal that is called MIMS, My Intelligent Machines, which integrates the data that I was showing you previously, the omic data, within an infrastructure of AI using deep neural networks. We connect that with environmental data, health data, depending on the clients that we are dealing with. To execute all this, we need to extract features. Features are derived mostly from bioinformatics and biostatistics, and those are mined directly from literature. Okay. This is one example where we can use AI to maximize productivity. The other one is annotating the human DNA. Okay. Everyone knows that human DNA is 3.2 billion characters, later ACGT, and we must have almost 21,000 genes within our genomes. Annotating the genome is really important, and knowing all the labels within our genomes and the other mammalian genomes are really important. One of the first projects I started in bioinformatics is doing Jurassic Park, where we have taken the mammalian genomes of almost 50 species, and we reconstruct the ancestral genome of their parents by using machine learning and estimating using Bayesian posteriors uncertainty based on that. What that can help? Using this genome and labeling the ancestral genomes, it helps us to better understand how the human genome functions, the mechanism behind. So then, with colleagues from Duke University, we explain better how the chromosome X is inactivated during evolution on the mammalian lineages. This paper was published in 2011 in Genome Research. So marking that you need to classify each character, each position within that genomic regions. That was predicting the back. Then we focus on saying, if we have our genome, I do have my genome, I just want to know if there are regions within my genomes that are fragile, that could lead to modification, and that modification maybe will alter next generations. So we design machine learning models to analyze, using the patterns of the different mammalian species, the predictions of different markers in the human genomes where there is a probability that a fragile site will be. So there is a kind of breakage that can happen during the evolution in that regions. So we went further. We analyzed viruses. Viruses that affect bacteria. Bacteria are important, right? Viruses also are important. So we construct models that use machine learning techniques to identify the origin of pathogenicity in HIV, in HPV, and also in phages. Just a brief example on how that works. Usually what we try to model, we mimic what we call the enzymes restriction fragment length. Enzymes, they cut the DNA. They cut the DNA in motifs. And those motifs, we can featurize those motifs and design a better machine learning models that can classify the pathogenicity, the functions, and deal with the recombination rate of some of the most virulent viruses. So several methods are out there. We continue with students of mine designing models that are based on alignment-free, using k-mers instead of biological fragments. We applied to hepatitis C, which is important in the pandemic now in Canada, and they are looking at a lot of relationship within the transmission between people, drug users. What we found is that using fragments, we can head in one other direction that are not headed yet. With the sequencing technology coming up, and real-time sequencing will be soon available at cheaper price, what can someone do is to embed it within the sequencing technology machine learning techniques that can directly detect pathogens that are present in the DNA. To detect that those pathogens, what you need to do is you need to find the most relevant features, and you need to associate that with the most important knowledge base that you have within your domain expertise. That's what we are dealing with in a paper that we just published in July. What that's helped us, those different tools that are based on machine learning, pathogen detection, fragment detection between pathogens, help us to design the first monitoring of pathogen within Senegal, that we are dealing with the Institute of Epidemiological Surveillance of Dakar, where what we have, we are monitoring the infection rate and the signature of the mutational landscape within the therapeutic effect. That helps to anticipate when drug resistance will be appearing to some individuals. And also, it will help to better understand double infections between HIV and, let's say, TB, tuberculosis, or other type of diseases. Those monitoring data are based on data that have been collected throughout the continent, and the patterns derive the infection and the pattern infection within the different African regions. So we can now better notice the subtype, the genotype, and the real type that are present within a single region during a time period of X. That will help us. We talk about annotation. We know the genomes. We know the positions. We know the targets, where the drug targets. And having those mutations, integrating with the monitoring system, will help us to better prevent within the healthcare. This is a paper that we just submit into PLOS Medicine. We went further. We used that monitoring system to identify mutational patterns between HIV-infected pregnant women during the different pregnancy phases to see how the mutational landscape is affected. So this also can be followed and tracked and used in model for better predictions. So in that case, we can go until to the proteins to see the combination between the surface proteins to see if there is a real binding within that mutation. So that platform is being constructed in Senegal. It is a fund that we have from the Ceramitic. It's a starting fund, but we will go further in that direction. The last example, several examples are based on agriculture. You can use the same position that I have within the biologists and the doctors, but now we can use the biologists and the farmers, or it's gone. We lost the sign. Okay. So let's just start from here. So the next challenges that we are facing in my lab is being not within the clinicians, but is talking to people in different areas: agricultures, dairy producers, people that make bread, and so on. They need to better understand the mechanism of the molecules that are present in the things that they are delivering. And also in the food chain industry, they are looking for pathogens. So we need to bring precision in that direction by using also the technology that I showed you previously. And that technology is not just integrating the omics data, but you need to combine the omics data with other type of data, including cameras from the landfills, sensors that are there to capture the growing of a plant, and so on. And those challenges are really important in the technology space, because knowing that you are in a land, there is no wireless connection, good wireless connection, you need to have a good technology to transfer the data. How to transfer the data into a secure location, how it will be treated, and how to bring feedbacks and bring the information right back to the farmers. Those are bottlenecks that people are facing within the precision farming industry. it's not only about the sensors and the cameras, but also what you need to capture. You need to capture all the other influencers of your plants. Let's say the CO2 affecting the lighting, the oxygen, the temperature of the soil, the nutriment that you have, and the microbes that are present in the surface, even down and up, because you can have both microbes, and how those different elements can influence the growing, the disease, and the productivity of a plant is important. There are a lot of people in the animal space or in the plant space working in that direction, and there are a lot of companies that are built based on that. Here, I will focus on cows and cereals, because let's just suppose one good thing about my job is to have these kind of things right in my lab to extract metabolites, but don't say it to nobody, okay? Actually, I will talk to you about something that is more serious than that. We all consume wheat, not weeds, wheat. Let's be clear about it, okay? So in my lab, we develop machine learning models to identify regulators of winter wheat. What is winter wheat? Canada is cold, right? And there is a wheat in Canada that you can seed in September, and it flower only in April after winter. The mechanism of survival during that period is not clearly defined. Capturing all the regulators that are involved in that mechanism can help to better control the growing of the plants, but not only that one. You will see later why. So we designed a complex machine learning models based on thousands of features extracted, and we found that there were 199 regulators that are really important in different conditions of the plant growing, and that can be used directly within the laboratory to enhance the productivity of that plant. There are tests that have been done by different groups to control the growing, depending on the regulators that are used. Because those regulators affect different modules, different proteins, different pathways within the plant growing systems. Okay. So the paper was featured. I told you there will be French, so here is. So why that is important, having those regulators in cold? Because last year, when I was at the global gathering of the Next Einstein Forum, there were the chef, Cham, who was talking about fonio. This is his slide, it's not mine, it is his slide. He brought 3 slides, and I took them all, because he said that fonio is one of the next gold of Africa, because it can grow, the growing is 2 months, and it can grow in regions that are difficult with less water and so on. And so you can have different records within the same year. So what we did since last year, we designed a group, what we call Les Amis du Fonio. And within that group, we are trying to identify the same regulators and see how we can enhance those productivity. So there are people in my lab also focusing on identifying metabolites. Those are chemical compounds. Those chemical compounds are used in natural products and sometimes are used by pharma for different drugs. So we just highlight more than 100 new metabolites that have been found in fungi. The paper is, it has been written. We will release it soon. Lastly, we were fortunate, my lab were fortunate to have the privilege, I'm choosing the word because it's so sensitive. The dairy industry in Canada is almost $13 billion. Okay. And it's so sensitive that when there were the NATO negotiation between the US and Canada, one of the bottlenecks was the dairy, because it's two ways of dealing with dairy production. The advantage of the Canadian industry, mostly in Quebecers industry, within the dairy production, is that they are managing quotas between farmers since the 1960s. Since they are managing quotas, to manage efficiently, they are collecting data. So they collect almost thousands of indicators for each cow every 2 days since 1990. And now, with those data sets, production data set, genomic data set, and also health, they are using the help of machine learning and AI specialists to integrate those data to help the farmers to make better decisions on their productions, enhancing the quality of the milk, enhancing the quality of the health, the health lifestyle of the cow, and so on. So my lab is leading that project for those who knows Canada from the eastern part of Canada, including Quebec, New Brunswick, Nova Scotia, Île-du-Prince-Édouard, and so on. So let's say we are dealing with almost 2 million of cows, which are held in conventional and free-stabulated environment. In the free-stabulated, the cow can go directly to the dairy extraction. And there is a bulk tank who take the milk every 2 days. Okay? So we collect the indicators from those bulk tanks. But also, we do have sensors and other indicators taken each month for each cow within the facility. So, our job as bioinformatician here is we have the production. Okay? So we know each cow from since 1990, each year, what he produced. We know his genetics. We know the farm where it was. We know the food that he was consuming. And also, we know the disease that he has been affected during his lifetime. So, can we integrate those data so that we can help the farmers to better manage their cows and their farms? Because both are important, the cows and the farms. I can manage my cows and I can manage my farms differently, because you can have a genetic that is poor, but you can be sufficient with some productivity. In the other hand, you can have a genetic that is rich and you have a better productivity. So there is no direct link between genomics and productivity, and dairy production, because there is the influence of the management, the health, and the other factors. So integrating all these layers are really a complex problem that we are dealing with for the next 3 years of the project. So what is surprising, the data collection and the health record of each cow is better organized than an electronic health record of a patient. Okay? And in the other hand, we do have 6 generations of individuals within the data. So you can use one part of the individuals, 2 generations, 3 generations, and predict what will affect the 4th, 5, and 6th generation and map those information. So we are in the process of distinguishing two herds. So this is one farm, the other farm, and depending, you can see how the productivity can affect. So the equilibrium for one farm will be 45 months, and the other one will be 35 months. So knowing the genetics and knowing the environment, you can anticipate those events and give better decision counsel to the farmers or the counselors that go and meet them. I think you are full of the genomics stuff. To that end, we are dealing with the genomes. We are using, for each position conformation, a markers. We have almost, it's a gene chip with 700,000 markers that are present, and those markers are related to, I will show you later, to different things. My lab is designing the platform because those data is sensitive, because if they are released, they will, for a farm, they will decrease their productivity, or they will kill the market for them. Knowing that there were some kind of disease in one farm will affect the whole farm. So in that kernel, we are extracting knowledge and transforming them into 3D technology to represent in three-dimensional space. We are looking for patterns, and we are building AI predictors for decision-making in the report. Every expert in the room is invited to join in the challenges here, because the challenges are in different directions. First of all, let's take a simple example. You give a farmer decision to say, you can keep this cow 1 year extra in your farm. It will produce more. That decision will affect the other decision that farmer will take the 2, 3 next other years. How do you model that in an environment where those cows also are in competition, because the space in the farm is limited? So if it keeps it, he will have to remove another one. So that environment will be to deal with. And I was saying that the genomic are direct related. I don't know why my slide is showing like this, but the genotype data are directly linked to conformations, to dairy power, and it associated to the structure of the legs of the cow. All those things are captured and used in the model for the prediction. So then we finish by having what they called a lifetime performance indicator, and those indicators will follow each cow during its daily life. It is a nice data set to work with. However, there are a lot of challenges where when we take each line, vertical line here is a cow, and the height is the month after birth, so the age of the cow. Okay. What we are saying, we cannot align all the cows because there are cows that are missing data. There are cows that are always caught at age 4. You don't know what the decision that have been taken for that. There are cows that haven't been monitored, or actually they were born in year 2, not year 1. So dealing with those data is really difficult. So my lab and with different animal science lab are working in that direction. And we are also working on integrating sensors and images within those farms to better manage the welfare of the cows, to detect condition, well-being, and so on. So it is important to deal with challenges of data collections. There are a lot of ethics issues in there. We are dealing a lot with the ethics. Data protection and privacy. Definitely, in this field, you need interdisciplinarity. So that's why the groups that I'm leading is composed of animal scientists, biologists, doctors within the same lab so that they can interact and provide a better models with the team of CS. Okay. So I will finish by my 2 dreams that I'm working on, actually. I have one friend of mine in Guinea who developed this amazing platform to capture sensors from data from patients. And we are building a machine learning models behind so that we can accelerate what I told you previously, the clinical triage, so that we optimize the relationship between patient and the lack of doctor that we are facing in Guinea, maybe somewhere else in West Africa. And we are in the process of patenting what we call an efficient and energy less consuming autonomous precision farming data collectors. That has been built in the serre where we grow our different cereals in Montreal. So those are my 2 main dreams that I'm focusing for the next few years. And tuning us throughout the services, it is a good challenge is to have the services to be digitalized, to have the online services accessible, dealing with electronic health, precision in health, precision in agriculture, and one last question is food safety. All those things are important regarding the use of machine learning and AI. It's very hard to combine, but it's a track where we don't have the choice to go. So I would like to thank my lab, the founders. They used to eat a lot because they consume a lot of energy there. Thank you.
Yeah, let's give Abdoulaye another round of applause. And if there's questions, there should be roving mics. Okay, and Shakir is gone at the back there. So you could just raise your hand and a mic should get to you. I'll let Abdoulaye run this anyway. So, yeah.
Okay.
Any questions?
Thanks. Thanks so much for the talk. So the question I have is, it seems like you're gathering this like richer and richer omics data. So you're getting these like massive, massive feature spaces. But the rate at which we're gathering maybe the label data is quite.
I'm a little bit.
So is that the right volume there?
Mm-hmm.
Cool. So you're gathering all this omics data, and you're getting these rich feature spaces that are just, as you seem to be growing in time. What about the bottleneck of gathering the actual label data of, clinical results, for example? So with the cows, it's great because they're tracked very well, like you said, but with like electronic health records, maybe they're not as complete. How do you handle that mismatch between the growth in the feature space and the growth in the quality of the labels?
That is a great question. We deal a lot with domain expertise here, where we have a lot of data that are labels. And we do a lot of curations within our fields because the decision that we need to take is really important. So in the process, we design tools to better curate and accelerate the curation of the features and the data within that space.
Hello.
Hello.
I want to ask you about the ethical challenges you've been facing, and since we know this data is quite sensitive, what processes have you been deploying to avoid like data breaches, that kind of stuff? Thank you.
It's really challenging to fill all the requirements of the ethics, but one reason that we had that grant is because of the guess that we made, we said that we will try to build an infrastructure that are robust, and within the area of dairy productions, there is a way of controlling the information so that it cannot come in the public space. And we will be going through those lines. So that is one reason I couldn't present some of the results that we are having directly within the sites here.
Thanks, Prof. The talk was really, really good for me. I actually just wanted to ask you, with regards to precision medicine, have you done any work on admixed African populations? Precision medicine for admixed African populations, and if you've had any challenges that you could speak about?
No, I haven't yet.
Okay.
But
so
I will
okay,
definitely.
cool.
Sure.
Thank you for a wonderful talk. My question is, you have started this collaboration in Guinea, and do you think you can do something about dairy, because there's a lot of also cows in there?
I'm a Fulani.
I know.
Actually, there is a joke about that. When I got the grant for dairy productions, I received a mail from Hamidou Tambine. He told me, "Hey, Fulani guys, how could you manage to get cows in Canada?" But seriously, it is really important to have those kind of transfer. We are discussing with an entity that is called Socodevi in Quebec, which is specialized in transferring technologies and scientific results from Quebec to other places in the world. And there is 3 phases, right? One thing that I found really, really amazing, it is the fact that in 1990, in Quebec, they decided to organize and collect and formalize the data collection without knowing what's going to happen 25 years or 29 years old later. I think that expertise first will help to better manage the different farms that we have, make a better selection, because I heard I was listening to the news in Guinea, and the president was talking about the other AI. Everyone knows what is the other AI? Artificial insemination. Okay? That is the other AI. Okay? So they were talking about inseminating cows in different lands, but there were no previous studies on genotypes that are present in there, the effect of the insemination, the relationship, the production, and so on and so on. Things that have been done somewhere else that can be captured and brought there to help better manage that kind of procedures. I think we will be in that phase, not in the phase of precisely deciding which cow to keep or not to keep within the next 10 years.
Hello, I have 2 questions for you. The first one is, you showed us a genome-wide association study, so just wondering if there is a better ways of doing such studies, because those require like a lot of people. So would you know any kinds of studies that would like be as good as GWAS? And the second one, I think there was a debate about how many cells are there in the human body and the number of microbes. Like, which side are you on? Like, are you on the side of there are many microbes, or
I am in the side of the microbes.
Okay.
I am in the side of the microbes. For the GWAS thing, Actually, in the data that we are dealing with, 2 million cows, we don't have that problem of the size of N because the population is really huge within that field. But when doing association studies with a small data set, you need to do a better design of your statistical framework. There are tools out there to better control in that space. We can discuss that if you want.
Hello.
Yeah.
My question is along the daily power in daily farming. Is there any way you can, like, or is there any solvables that you can come up with, which is, do you have, okay, in your lab, do you have any solution for improvement of production? Like maybe a natural way of cows like some liters and you want it to boost of 40, 50, 60. Is there any solution that can actually, is there any solution to that?
I missed the first part of your question. Can you just repeat that?
My question is of daily farming. And on the genotype of what you are doing in your lab, is there any? Do you have any solution like that you have come up with which can boost up the production?
Yeah, actually, the results we are having, we will be releasing them to the farmers soon. By better managing the selection of the genome, the genotypes that they have, can increase for 20 or 30% their productivity. That's one step. But that is really difficult to control because you will have it during time. Because you decide the insemination before having the production. But the other, there is a 25% that is really affected by the food that the cows eat. The mixture, the timing, and the timing of the nap in the farm. Those different indicators contribute to 25 to 30% of the productivity. The other ones are the health. Mostly the health is difficult to control. For now, we didn't look that much in that direction because we still in the process of discussion with the veterinarians to handle efficiently their data within the framework. And we are also dealing with the ethics in that direction. Actually, we are at that level.
Good afternoon. In your models, I saw you used, you said some of the models you used are stacked autoencoders. Can you just explain how you use that?
In fact, when we use the stack, it's when we integrate different models, the data coming from genotyping is organized within linear sequences, and the expressions is numbers. You have expression numbers and alternative numbers that are out there. Combining those things need to have a better combination on your neural nets aspect. That's why we use the stack. Okay.
Yeah. Thank you again, Abdoulaye. All right, that is the end of this session, the keynote. Just a couple of announcements. One, Kevin Kimayo, if you lost your ID card, please come see me on there. Two, if you are yet to register you are requested that immediately after this this session ends, to please go to EBR 30, so go behind, and please get your registration sorted out before continuing on with the afternoon sessions, and then I know there'll be coffee and tea right now. So just to also highlight, again, the sessions that are happening at 4:30, it's going to be convolutional models in SCZ 39, which is this room. There'll be generative models in EBR 30.